see you and you can see them, and then there's people online,
okay,
and they can see the things. Well done. There's no snow, so there's not a lot of reasons why everyone's late, but it is Friday of what felt like a very long week because of the snowstorm. So welcome everybody to Friday, and thanks for being here. I'm really excited to have our first pathology talk of 2020 by Dr. Shreeram Akilesh. He's a assistant professor at the University of Washington in Seattle, and actually drove here so as not to actually get caught in weather and funny planes. But importantly, Ram's made a number of fundamental contributions to understanding proteinuria, the genetics of nephrotic syndrome, and the cell biology basis of the podocyte. And so we don't often have the opportunity to have a pathologist talk to us about structure and function of the kidney genome, so it's really wonderful to have you here. And as you talked about with the fellows in the Jeopardy game that you guys played yesterday, I want to know who the winners were. But I think it's important as we enter 2020 to remember how much we can learn from biopsies and looking at things, and I think this is the beginning of a new era in nephrology, so really thank you for helping us understand better. So welcome.
Thank you very much, Adira, and Mazi as well for the very nice, kind invitation. Can everyone hear me? Great. So even though I'm a pathologist, today I'm actually not going to show you a single biopsy slide. Okay, so I'm going to tell you more about what really drives me to think about when I actually look at biopsy slide and you look at the cells and you look at the nuclei, what's really driving them to have the phenotype that they have? What's then allowing them to act in aggregate together to result in the organ dysfunction or normal function, and ultimately the clinical presentation for our patients. And can understanding the genome and the mechanisms that are driving the cells to respond and react to different kinds of insults and injuries and stimuli actually give us new insights into trying to understanding disease mechanisms and processes and ways to actually intervene. Okay. So some of the topics I'm going to talk to you today are really going to take across a wide swath of things. We're going to talk about kidney genetics. I'll even talk a little bit about kidney cancer, which is actually topical since a fellow nephrologist actually won the Nobel Prize last year, Sir Peter Ratcliffe. And lastly, I'll introduce some new unpublished work, which I think is going to be very exciting in the field of cell-free DNA. And I'll try to connect these together in terms of the basic biological studies I'm doing in terms of epigenetics and epigenomics, and how that can actually translate into real, tangible insights and potential interventions for our patients. Okay, so I have no financial conflicts of interest to disclose. So just by way of introduction, I'll introduce to you the topic of what I mean by epigenetics or epigenomics. This is kind of the modern definition of this term. And then we'll go through 3 different vignettes. As I mentioned before, I'll talk a little bit about kidney cancer here. And then I'll talk about structure and function of the DNA in the nucleus of the cell and how that actually contributes to the cellular phenotype. And lastly, I'll talk about unpublished work about defining molecular biomarkers for kidney injury. So let's begin with what I mean by the epigenome. And so the classic definition of epigenetics really means a non-sequence-based trait that can be inherited or passed down to the next generation, the daughter cell or the organism. Okay? In the modern term, this term is actually really conflated to mean anything that is not directly gene expression-based or directly in the DNA sequence itself. So it involves a variety of modifications, say, of histone proteins and other regulators that control how the genome is actually working. Okay? So it's all the stuff that is outside of the genetic sequence itself, but that's acting on that genetic sequence to interpret and derive that information to then produce the cellular phenotype. Now, if we think about this, each nucleated cell in our body has in its haploid genome 3 billion base pairs. If you were to stretch this end to end, this is going to be about 6 feet or 2 meters tall, about as tall as I am, right? Squished into every single nucleus of each cell. So you can see that this is a tremendous physical burden that the nuclei of cells have to contain. So the way they actually deal with this is by wrapping most of the DNA around nucleosome protein particles and even higher order structures such as chromosomes to keep all this DNA compact and jammed into the nucleus. But this, of course, presents a problem. Okay? So if the cells actually have to turn on a gene, they have to find it in this very compacted mess. open up specific regions, and then read that DNA sequence, that code underneath, to then interpret and execute the genetic program, if you will, for that cell. Okay? So there's a problem of the fact that the genome is generally repressive for gene transcription, and the fact that the cells have to find a way to transcribe those genes that are important for their function. So the way this happens is that there are a variety of machinery, protein machinery, that interpret and act upon this compact nuclear chromatin and open it in very cell type specific ways to then execute the cell's desired genetic program. This could be in response to its normal differentiation. It could also be in response to external stimuli that then are funneled into the genome and ultimately changes in gene expression. Now, the key point I want you to remember and to take away, one of the few things you take away from this talk, is that these patterns of opening of the nuclear genome in specific locations are extremely cell type specific. Okay? They show specificity at the level of not only the cell type, but also its context, its previous exposures to drugs and medications and various insults, and may even have predictive potential in terms of how it's going to respond in the future to additional injuries. Okay? And many of these features, in terms of, say, histone modification, CPG methylation, and other regions of the genome which are open in certain cell types, such as enhancers, are all upstream of gene transcription. So even before an mRNA transcript, a gene is transcribed, we already have these marks in the genome that the cell is sitting and poised and ready to respond. Okay? So let's show this schematically. So again, as I mentioned before, most of the nuclear DNA is very compact around these nucleosome and higher order structures. And you can see these genes that are represented by these little cylindrical marks. If we were to say, how is this particular gene, which is now in its off state, actually turned on? So our current understanding is that when genes are activated, this requires transcription factors to bind to specific regions such as enhancers, which requires them to actually displace the nucleosomes off that compact chromatin, open up the DNA, and then bind to the DNA and read what's going on underneath. This is also associated with a physical reorganization, so that distant enhancers, which may be hundreds of kilobases away from their target gene, are actually brought in physical proximity with their target gene, and this corresponds to activation of gene transcription and production of mRNA that, of course, we can detect by a variety of means. Okay? So this is our current understanding. So it requires multiple things to occur. It requires a reorganization of the chromatin, displacement of chromosomes, binding of transcription factors, and physical reorganization of the DNA within the nucleus to actually turn on genes. So all these epigenomic methods really are trying to understand this process, and like the blind man, kind of feeling around the elephant, they each give a part of the picture, but not the complete picture. So most people have probably heard about RNA-seq. This is kind of the modern way to detect and measure gene transcription. That, of course, is going to measure the production of mRNAs and, of course, many other type of RNA moieties as well. ChIP-seq might also be familiar to many people here. So this is where we actually use antibodies that recognize a specific motif. So I've shown here, for example, a variety of histone modifications, but it could also be certain proteins that bind to the DNA or any other protein complex that is interacting with the DNA. It can be labeled with this antibody, pulled down, and then the attached DNA can then be sequenced and mapped to the genome to identify where in the genome this particular modification is labeling. And a third entity, which is going to be a feature of the talk I'm going to for today, is methods that actually identify where these open chromatin regions are. These open enhancers that, as I told you already, are very cell type specific and are driving an upstream of the transcriptional response of cells. So two common methods are DNase-seq and ATAC-seq, and these are really, again, saying where is the DNA open in contrast to where it's compacted around nucleosomes. Importantly, this is not the same as genome sequencing. This is not going to tell you the linear sequence of genome. It's actually going to tell where in those 3 billion base pairs some regions are actually open and being interpreted by that particular cell type. And lastly, there are methods to actually measure changes in the physical conformation of the DNA. Again, it's not a linear sequence. It's kind of, if you can imagine a bowl of spaghetti, that's really what the DNA is looking like in the nucleus. But that spaghetti is roiling around. And that roiling around is different from one cell to the other. And these methods, such as Hi-C, are sequencing-based, can actually allow you to tell which regions are actually touching each other in the genome. So let's go in a little bit more detail methodologically about how DNase works. Again, as I mentioned, this is an enzyme that actually cleaves DNA. And since DNA that is already open is much more accessible to the enzyme, the DNA fragments are preferentially released from these open enhancer regions. Of course, they conveniently fall into the nice size range for next generation sequencing, and that's what we do. So what does the data actually look like? So if we were to map all these individual DNA fragments and see where they line up on the genome, and see where's the histogram, where do they pile up more often than others, we get profiles such as these. Okay? So I'll walk through the key features of open chromatin profiling, such as DNase-seq and ATAC-seq in the next couple of slides. So what we're seeing here is if you were to take, again, the genome and stretch it out in a linear fashion, each of these correspond to a nucleotide level resolution of that DNA sequence aligned across the genome. Over here, we have a little model of a gene. In this case, it happens to be WT1, a known podocyte marker. It's running from this direction from left to right over here. And these tall hatch marks represent the exons, the protein coding portions of the gene. The thin line in between really represents the introns, the non-coding portions. So one of the key features you can see is that those small DNA fragments, as they pile up into a histogram, result in these little peaks, which are actually mapped to a precise genomic position because of the DNA sequence that's inherent to them. So what are some of the key features? These little peaks are known as DNase hypersensitive sites. For this talk, we're really going to be kind of saying that these are equivalent to some kind of gene regulatory element or enhancer element, if you will. This is an element that we are saying has some information that is controlling genes that may be nearby or far away. One of the key features is that if properly done, properly executed, these can be extremely reproducible maps from patient to patient. So here we're showing 3 different tracks from glomerular cultures from 3 different patients done under different time points of collection, but still you can see that the overall pattern is extremely similar. And that's the same thing for the tubules here from another 3 patients. The converse of that is that each cell type may have a very unique signature. So you can see that while within a particular cell type or sample, the signature is very similar, you can see that across cell types, for example, this particular peak is only present mostly in the glomeruli, a little bit in the tubules, and not so much in these other cell types that are shown here. And conversely, there are other peaks or enhancers that are present in other cell types. Okay? So now I'm showing you a very small region of the genome, okay? Probably only about 100 kb or so. So think about the 3 billion base pairs. We have this information now mapped across the entire genome for multiple different cell types. And these individual peaks or enhancers in composite form a unique signature for the regulatory status of that particular cell. A third important point to remember is that the majority of these peaks are actually very far away from the promoters of genes. So again, if you were to say that this is the beginning of the gene, this is where transcription begins and goes away, sure, there's open chromatin here, but the majority of the enhancers are actually quite far away. And this is, again, providing information that even though only 1 to 2% of our genome actually encodes for protein coding genes, the rest of the genome contains these regulatory switches and enhancers. And these methods actually allow us now to identify exactly where they are. Okay? So you might be saying, if you're a nephrologist, how does this actually apply to me? How can we actually extract information from this epigenetic these maps that are even upstream of gene transcription? So let's go into a few vignettes to try to make this very concrete and tangible. And we'll start first with kidney cancer. Okay? So let's...
Whoever is moving things around, can you just mute yourself, please? So let's start with the first story. So I mentioned again that to open the chromatin, we need transcription factors. Okay, so these systems are clearly a very good way to understand how transcription factors actually affect regulation of genes. So let's start with this first model system, and that is kidney cancer. So the source of the materials in these studies are kidney nephrectomy. So here is a kidney, an entire kidney, that has been removed and bivalved or cut in half
exposing this kidney tumor, clear cell carcinoma in this case, and abundant, grossly normal kidney tissue in the background and surrounding fat. So this is really a nice abundant source of material since some of these genomic methods are very tissue hungry. They need a lot of input material. But we can take the cells, say the kidney cancer cells out. They can grow out in culture. We can take kidney cortex and tubules as well. They grow out really well. You can see that they look quite different. The cancer cells actually grow much slower than tubule cells in culture. They also oftentimes have vacuoles, which are typical for adenocarcinoma in culture. So let's start with what is known about transcriptional regulation in kidney cancer. So this is, of course, the topic of the Nobel Prize for Sir Peter Ratcliffe, Greg Simanza as well last year. So kidney cancer or clear cell carcinoma is canonical for hypoxia signaling response, right? So under normal oxygen conditions, the tumor suppressor gene VHL constitutively ubiquinates and degrades the hypoxia-inducible factors, which are transcription factors that will become important in a second. Under conditions of hypoxia, this normal constitutive degradation process is interrupted. So HIF proteins are stabilized. They go into the nucleus and they activate a slew of genes which are adaptive to that hypoxic response. Okay? So they'll turn off VEGF, that's promoting angiogenesis. They'll turn on EPO, promoting increase in red blood cell mass. They'll change expression of glucose transporters to allow cells to actually survive under glycolytic conditions. Now, what happens in renal cell carcinoma by a variety of methods is that VHL is genetically inactivated. So these tumor cells behave as if they're constitutively in a hypoxic state. So HIF is constitutively activated, and this gene transcription program is jammed on. Okay? So this is what was known at the time that we started this study. So again, as I mentioned before, we could get these samples out. We actually generated our epigenomic maps, and this is just a kind of the integrated approach, a schematic of what we did. So as I mentioned before, we generated these open chromatin profiling maps. Here's a gene called stanocalin-2. It's often upregulated in kidney cancer. And if you compare the tubule versus the cancer now derived from the same patient, okay, so we're controlling for genetic background more or less here, we can see that there are many open enhancers that are activated, which are in the tumor, but not in the tubule. Okay? Now, the reverse is also true. In other regions, there are enhancers that are open in tubules, but not in the cancer. Corresponding to this, we can also see gene transcription, corresponding to these higher histograms in red in the tumor compared to the tubule over here. We can now integrate this with other external databases, such as evidence for HIF binding at the promoter of this gene, or also large-scale tumor gene expression databases, such as the Cancer Genome Atlas, where they see on average that tumor cells are overproducing this particular gene, to really kind of integrate these various types of information to try to glean insights. So we took this approach to say, let's look at these hypersensitive sites or enhancers that are showing differential activity between tubule and cancer in either direction. Okay? Can we understand something about the transcriptional regulation of this cancer that goes beyond the hypoxia signaling canonical information? So remember again that in these hypersensitive sites, if we were to look very carefully, we can actually see the DNA binding motif of a variety of transcription factors. By the very fact that we know where these sites are, we can examine the sequence underneath them and identify which transcription factors may actually be binding at those locations. Okay? So doing this analysis, we can ask which enhancers are more active in the cancer compared to the tubule, or vice versa, or which ones are really not changing between tumor and normal. Okay? And examine underneath there which transcription factor binding sites are enriched or de-enriched in either condition. Okay? So this, of course, shows a number of findings. For example, HIF is actually in kind of non-changing enhancers. Okay? Again, saying that it may have a much more complex role than just in an activating role that has been commonly understood. But there was one particular transcription factor motif that we were particularly interested in because this particular gene is also known as OCT4. You might be saying, what is OCT4? Okay? So OCT4 is one of the 4 transcription factors that when put into somatic cells can actually reprogram them into stem cells. So this is another subject of a Nobel Prize as well. And so we were very curious as to why a stem cell, a very potent reprogramming factor, was being consistently enriched in the enhancer landscape of kidney cancer. So we went back and asked, in our tumor samples, we saw that consistently OCT4 was actually upregulated compared to the tubule samples. No other family members were actually upregulated as well. And then if we look at a much larger cohort, almost 500 tumor samples from normal kidney versus matched normal, from kidney cancer versus matched normal control, you can see that the OCT4 gene is actually upregulated fairly consistently in kidney cancer, actually also in papillary cancer, but not in a variety of other epithelial malignancies. So what have I shown you so far? We've known so far that HIF is very important in kidney cancer. By generating these enhancer maps and by understanding what transcription factors may be binding at those particular regions, we can ask which transcription factors may be differentially active or inactive in kidney cancer versus normal tubules. Okay? From that analysis, we identified that maybe OCT4 is actually having an imprint on the enhancer landscape of kidney cancer. And we're actually able to connect it to increased expression of this potent transcription factor consistently in large tumor registries. So the next slide is complicated, but let me walk you through this, okay? Because it's actually going to tell you a very interesting evolutionary story in addition to the mechanism of why OCT4 is upregulated in kidney cancer. So let's step through this a very integrated approach here. So here we can see the gene model of OCT4. It actually has multiple transcript isoforms. Here's where the ES cells actually activate transcription of that gene. When we look at the gene expression profile, so RNA-seq from tubules or kidney cancer, we can see that there's actually a very long transcript that originates far away from the normal start site of OCT4, many kilobases away, which actually corresponds to an open chromatin region, an enhancer, which is present in tubules and kidney cancer, but not in embryonic stem cells. Okay? Interestingly, this particular region, which is acting as a novel promoter for OCT4, is only present in humans. So you can see this gap in the green bars over here means that this region of the genome is actually not present even in chimps or our nearest ape relatives. Okay? And it corresponds to a recently inserted retrovirus that carries a HIF binding element. So let me walk through the story again for you so you can understand this. So what I'm saying is here, a lot of our genome has endogenous retroviruses that have been inserted and which contain very potent transcriptional elements. Our genome has found a way to silence the majority of them. This particular retroviral element is present only in humans and carries a HIF binding site, the same HIF binding site that is constitutively activated in kidney cancer. Okay? So now we have a connection. We know that HIF is constitutively turned on in the majority of kidney cancer. It now can find this Trojan horse of a HIF binding site in this particular element to drive OCT4 expression from a novel promoter. Now, interestingly, a lot of other animals actually don't get kidney cancer. And so this is, I'm wondering, for example, the mouse is a terrible model for kidney cancer, actually. And so I wonder if this is also part of the contribution that this very potent stem cell factor is kind of loosening the genomic environment, allowing kidney cancer to evolve in human beings. Now, this increased level of transcription also corresponds to patchy expression of the OCT4 protein. And importantly, the transcript levels actually also have an effect on prognosis, meaning that higher OCT4 levels, that red line over there, correspond to poor prognosis in patients in the Cancer Genome Atlas database. So what I've told you so far is really a story of transcriptional dysregulation. Again, as one example, one application of these epigenomic methods. We know that HIF is important, but HIF can actually bind and have secondary effects, as is the case in this particular story. Here it found an endogenous retroviral element that carried a HIF binding site and turned on OCT4, which is a potent stem cell and reprogramming factor. with an effect on the entire enhancer landscape of kidney cancer. So this is just one example of how we can understand transcriptional dysregulation using these epigenomic maps. And so we've done this also with normal kidney as well, and we can identify a variety of signatures such as enhanced STAT signaling and IRF signaling and HNF signaling as well, which I don't have time to go into for this particular talk. So let's now switch back and go into native kidney and see what can we understand about normal kidney cells such as glomeruli and tubules, and can we understand something about how population genetics can actually be brought into these epigenetic maps. So we did these same methods where we can actually isolate cortex, physically isolate glomeruli. When you lay them down on a dish, the podocytes and mesangial cells will grow out. And again, we can take the cortex and grow out mostly tubular epithelial cells. These generally have the phenotype. The glomerular cultures have a kind of a mixed culture of mesangial cells and podocytes. Endothelial cells generally don't survive in this particular culture. And then the tubule cells also have a tubular phenotype, albeit under these conditions they have kind of a little bit of an injured or slightly dedifferentiated phenotype. So again, looking at what the data looks like here, again, here's WT1. We can see that this is a known podocyte marker, and it shows nice, in this case, gene expression, RNA-seq, in the 4 patient samples that we had. Again, remember that these were done under different time points, but there's all this talk about batch effect. But when done properly, this can be extremely robust and reproducible. So as expected, we can see that the glomerular cultures have nice expression of WT1, but not the tubule cultures. And again, looking at the enhancers around that, while there's activity around the promoter of WT1, all these red arrows here are pointing to the enhancers. So this is now the map of distal regulation or control of how WT1 is actually expressed in podocytes, which had not been described at all before. So how does this actually intersect with population genetics? Okay, so here I'll introduce the concept of GWAS, or genome-wide association studies. The basic principle here, of course, is that if you were to take 2 cohorts, say a healthy cohort and a disease cohort, or a cohort having some phenotype that you can measure, and then you were to ask what's different between these 2 at a genetic basis, if you were to genotype them, you can actually find genetic regions that are more likely to be associated with healthy or disease states. And then you can investigate those regions to say what genes might be here that may be contributing to this phenotype. Okay? So this, of course, has been applied to thousands of different conditions, measurable traits, and phenotypes. And in the last 10 years or so is also now gradually making its way into the kidney field as well. And I will say one pitch for this is these are only as good as the markers being measured, right? So creatinine is a terrible measure of kidney function, right? But many of these GWAS studies are based on that. So we'll keep that in mind as a caveat as well. Now, one interesting finding, the way these genome-wide association studies are actually performed, is that they're looking at single nucleotide polymorphisms, or SNPs, across the genome that are variant among individuals. And almost uniformly, what was found is that regardless of the trait or phenotype, the majority of these genetic risk factors, these SNPs that are associated with diseases or traits, are not in the protein coding regions. Okay? So this is another important point I want the fellows to take away as well, that you can get a lot of information about genetics from these genome-wide association studies, but almost uniformly, the high-risk variants are outside protein coding regions. The notable exception, of course, in the kidney space is APOL1, which has coding variants in them. But this is an important point. So then how do these risk variants actually contribute to the disease that's being measured? Okay, so that was a real puzzle and a mystery until about 10 or 12 years ago, when it was found that the majority of these risk variants, which are outside protein coding regions, actually concentrate in those very same enhancers that I was telling you about that are controlling cell type specific function. Okay? So it was found that these SNPs that say are associated with heart disease are more likely to fall into enhancers that are only present in heart cells or cardiomyocytes. Okay? And so this is again providing the link between genetic risk, which is scattered across the genome, is really pointing to enhancer elements that are extremely cell type specific. which now provides a connection between genetics, population-level genetics, to actual dysregulation of genes in a very cell type specific fashion. Okay? So how do we actually get to this? How do we actually then connect a risk variant or a GWAS SNP located in an enhancer to a particular target gene, which could, I mean, there are many potential targets over here. So we can use this measure of physical proximity as well to say that this enhancer is now brought close to its target when it's activated to identify a potential target gene. Okay? So let's step back again and say, how are we going to actually apply this to the kidney? When we started these projects, many of these maps, which can measure these different viewpoints of the genome's function, were not present for the kidney. And so we went about generating open chromatin maps, such as DNase-seq. We have generated RNA-seq maps from glomeruli and tubules. And then more recently, we generated these physical proximity maps, which tell you which 2 points in the genome are actually touching each other. Okay? So we decided to put all this together to really see, can we identify what are the real potential target genes of these genetic risk variants for kidney disease, and annotate what their target genes might be to really give some insight into what new mechanistic pathways are present. So again, in word form, what we're going to do is look at the enhancers that are present in glomeruli or tubules, look for kidney GWAS risk variants, look for changing genes, and look for contacts, and really intersect them all. Okay? So most studies use only one or two of these methods. We wanted to use all four. So we wanted to be very stringent about what we called a target gene for a kidney disease risk. And so really we are taking epigenetics, intersecting with population genetics, understanding gene expression, and also actual physical conformation of the genome to do this. So here let's take an example review of how we did this for one target gene. This is CDKN1C, also known as P57, another podocyte marker. So here what we're looking at is what's known as a genetic association plot. And this is, again, the results of how these GWAS studies actually look. The higher the dot is, means that this particular variant, or SNP, is more likely to be associated with, in this case, CKD. Okay? And so this is pointing to a particular region of the genome which is highly associated with chronic kidney disease in this particular population cohort. If we actually look, there aren't very many genes around here. There's a non-coding gene. The nearest protein coding genes are over here, such as the CDKN1C. So how is this variant actually affecting gene expression? That was the question, right? So we can then look and say that, oh, look at that. This particular variant lies in an enhancer element that is actually more open in glomeruli versus cortical tubules. The only gene in the vicinity that is showing any change in expression is CDKN1C, or P57. So that maybe says, maybe this enhancer, this risk variant that's lying in that enhancer, may be regulating a P57. But how can we show that? So we can actually then look at the physical contacts across this region, and we can see that there are long-range contacts between this enhancer and the target gene. Okay? So really we're integrating multiple levels of information about how the genome is functioning to have high confidence that that particular SNP is actually interacting with P57 and potentially affecting its transcription. Now here's a corollary for that. So this was a genetic association study for chronic kidney disease. But this is annotating a podocyte-specific gene. Okay? And so what it's also telling you is that these mechanisms, if we identify a SNP that's present only in a particular cell type, it may actually give us insight into what cell type is also participating in the disease process being measured. Okay? Now, this is just one example. We applied this genome-wide using this very stringent methodology and annotated over 45 new target genes. So some of these are actually known, such as the ones in red arrows, but many of these are new disease pathways for kidney disease, which have now got much higher stringent levels of epigenetic evidence based on our data. We're actually following a couple of these related to diabetic nephropathy as well, using mouse models. Now, this has really opened up the GWAS field, and so more and more people in this field are actually using these maps and identifying that these SNPs, which they hadn't seen before, now fall into kidney-specific maps, which were not available. So here are a couple of papers that we contributed to recently. For example, this particular SNP was in the middle of nowhere, about 20 kb from any gene, and we can see again that it falls into open chromatin regions or enhancers in glomeruli and tubules, again providing a mechanism. And you might say, why does that actually make, what additional information does that tell you? So if you were now to then see if this is the target gene for this, you can look at the transcription factor binding sites here. You can look at all these others in the vicinity to see what might be the control that the cell is actually using to turn on or off this gene. You can see which other cell types this gene might be expressed. You can also look at the physical conformation of this to see how this whole genomic locus is structured. So it gives you very tangible information about how to then prosecute this candidate gene in a mechanism-based approach. So more recently, we're trying to take this even closer to the tissue. The data that I showed you before was using really cultured cells. And I told you right at the very beginning that these maps are super sensitive. So they're sensitive to the state of the cell. So clearly growing things in 2D is a non-natural state for these cells. They're grown in the presence of serum. That's also a non-natural state for most cells. And so here we have more recently generated maps from native kidney cortex and medulla. Gratifyingly, the majority, about 70% of the sites are actually shared with our previous maps. However, there's about 24,000 sites which are completely new and only present in the native tissues. Okay? So hopefully we're getting closer to the reality in terms of what's the real epigenomic state of these cells. This has already been fruitful because when we look at those sites and ask what are the genes around those regions that are actually annotated, it's related to solute transport, brush border integrity, and metabolism. So the things you might expect that may be lost in culture are actually retained in the native tissue. Okay? In addition, we've again cross-referenced our GWAS maps, and 17 additional loci fall into these new enhancer sites that we've mapped, including, for example, UMOD, which has always been at the very highest of all the CKD GWAS. But now we can see that there are 3 enhancer sites which were just never present in any of our previous maps. Now we know that these high-value SNPs actually fall in enhancers in cortex and in medulla in particular. So again, this shows us, this is a thrust for our lab, is to try to get closer and closer, try to isolate native cells and not culture them as much as possible so that we can get closer to the real state of these cells. In addition, I mentioned we were looking at chromatin conformation. So this is the map that we generated in our Jason paper last year. What we're looking at here is a contact frequency map across the entire genome. So each of these boxes represents one chromosome. So this is a very high-level view of the entire genome. So this is showing the contact at each point with every other point in the genome. Since that, we've generated additional maps from the whole kidney cortex and medulla. And again, I want to emphasize that these maps don't exist for any other native tissues, not even heart, lung, et cetera. We only have it for kidney now, all right? So these are, again, going to provide us new insights into how the genome is organized. And you might say, well, you know, this just looks like a diagonal of red squares. I mean, is it really different? You know, you spent a lot of money and you mapped 1.2 billion contacts, so what, right? But you got to look at, this is the 35,000-foot view. So if you take the renal cortex and medulla, down from the same patient, and zoom in on, say, any given chromosome, and you ask, are there differential structural changes? And then you start seeing fine structural differences. So here I've color-coded regions which have an increased contact frequency either in the renal cortex in red or medulla in blue across chromosome 18. So along the diagonal would mean equal contacts, but you can see that clearly there are regions which are showing higher organization and compactness in cortex compared to medulla and vice versa. And this is not the same. Some chromosomes look very uniform, others look very choppy and almost like a plaid pattern. So again, we are now trying to understand what does this mean in terms of actual physical structure of the genome and the underlying gene expression, which has never been done in any other system to date. So here we'll hopefully get an insight into understanding how actual physical conformation of the DNA is going to translate into controlling gene expression. So in this part, what I've tried to impress upon you is that we have generated these maps now for kidney under cultured conditions. We're moving into more native states. This really is a way to understand population-level genetics, because all those risk variants are really concentrated in these enhancers. And so by spending time to generate these enhancer maps, we're really trying to understand what's the transcriptional control and regulation of the target genes. By integrating these various methods, not using just the one blind man approach, but taking the whole team together, we can really have a much higher confidence in linking a particular genetic risk variant to a target gene. And some of these may be hundreds of kilobases away, skipping genes in the middle, right? And so allowing this physical conformation to be another factor increases our confidence of annotation. So in the last few minutes, I wanted to talk about an ongoing project which is related to using these same epigenomic maps to maybe get some insight into novel biomarkers for kidney injury. And so here I'll introduce the concept of cell-free DNA, which many of you might be aware of. And so the process here is that when cells are injured and undergo apoptosis, they activate enzymes to actually cleave their DNA. This results in that DNA laddering, which we've all learned about. But the fact that those enhancer regions, which I told you about, which are already open, they're going to be preferentially susceptible to this cleavage. So in the early stages especially, DNA is going to be preferentially released from the enhancer sites compared to the background of very compact nuclear chromatin. Now, this has already been leveraged to try to identify a cell of origin for tumors and to identify tumor types. But we wondered, now that we have kidney-specific maps, can we actually leverage this to understand kidney injury? Okay? And so again, the key point to remember here is that cell-free DNA really is going to be preferentially released from cell-specific enhancers, which are highly chromatin, accessible chromatin regions. So let's again go back to these enhancer maps. And here we're looking at 3 kidney injury markers, KIM1, IGF-BP3, and NGAL or lipocalin-2. And remember I told you that our initial cultured enhancer maps have an injured phenotype. Okay, so they were grown under 10% serum. We also have access to other tubules which were grown under 0.5% serum, and now we have our native tissue maps. So we were struck when we looked at these regions, such as around the KIM1 gene or HAVACAR1, that native tissue has very few open chromatin sites compared to the widely open regions at very distinct points around the KIM1 locus. Same thing happens around this injury marker, lipocalin-2, and there's a whole laundry list of these that we can go under as well. Interestingly, it's not only unidirectional. We can see that there are some enhancers which are only open and accessible in native tissue, but are lost in the injured state as well. So we have these regions. If we had some way to measure these regions, we could have a sensitive marker of kidney injury, in this case tubular injury. Okay? And so again, remember that since cell-free DNA is going to be preferentially released from this, we are now trying to actually measure and sequence using whole genome sequencing plasma cell-free DNA from patients with acute kidney injury in the ICU. And we can see when we look at a fragment size distribution, we see an enrichment in these patients in small fragment sizes, which is exactly where we would expect enhancer-derived DNA to be located. And so we have a cohort of about 15 to 20 patients right now. We're trying to analyze this a little further. But really, this connects the fact that we know where to look for those kidney-specific enhancers. We can now look specifically for those regions in the cell-free DNA of these patients to ask, does the acute kidney injury that they're manifesting clinically correspond to a cell-free DNA biomarker of kidney injury? So we're still in very early days on this, but we're excited that we've identified now regions across the genome that are showing that graded response of their enhancers up or down. And we now are looking at those particular regions in these patients with acute kidney injury, both initially in their plasma, but we also want to look at urine as well. So again, the last point just by summary here is that these small DNA fragments in the cell-free DNA are going to be derived from enhancers that may actually, because they're so cell type specific, identifying where they are itself might be the biomarker of which cell type is injured. Okay? So here I want you to radically rethink about what you think about biomarker, because here the genome itself, the specific location in the genome, becomes a biomarker in the case of cell-free DNA. There's always a lot of hype with these things, but we'll see how this one plays out. But I think there's enough traction in here that we might be able to get some positive forward progress. I've not had time to talk to you about some of our kidney on a chip experiments, where we can actually grow tubules in three-dimensional cultures. They actually become much more normal. With collaborators at the Kidney Research Institute, they've actually exposed them to serum and done gene expression. So now we can ask from a completely different orthogonal view what genes are changing in there and look at the chromatin landscape, and they again mesh very well. So unfortunately we don't have too much time to describe that, but this is really a nice cross-functional way to actually try to understand what are the potential genes that are changing, what are their chromatin landscape changes, can we measure those changes in the cell-free DNA as well. The chip system is powerful because we can then introduce precise insults, such as particular tubular toxins. We can introduce ischemia, hypoxia, et cetera, and ask what are the specific signatures associated with that. So you can imagine a time where we can take a cell-free DNA and maybe even parse out the particular contributions of different types of injury to that. So that's my 10-year, hopefully, pipe dream, but we'll see how we go in there. So I titled this talk Kidney Structure and Function of the Genome, right? And so as a pathologist, this is what I look at every day. And we're always thinking about kidney structure in terms of the biopsy that we see. And we try to correlate that with dysfunction that our patients might be manifesting. But I would say that, when I look at these biopsies, and if you zoom in and you look at the nucleus of a podocyte, or you look at a nucleus of a tubular epithelial cell, I want you to think about some of those topics that I just mentioned to you today, right? All that stuff, all that complexity, all that regulation, all that genetic background that we all bring into this is at play every day in the nuclei of these cells, right? And may be different from one cell type to the other. So I don't want to scare you with this emergent complexity. I don't think that that's the point. I think it's also beautiful that there is that much complexity and it still functions normally in the majority of patients. But I think by trying to use some of these precise tools to really kind of understand and decipher them in a careful, controlled way, we can really take it from the bench, hopefully all the way back to the bedside. And so I'll stop there. There's a lot of people I need to thank, of course, and funding sources as well. And we have a few minutes for questions.
The kidney on a chip stuff is has a lot of play. And I'm just wondering if you can explain, because maybe not everybody realizes, what you've talked about here and the kidney on a chip is sort of the way that we're going to understand better susceptibility to disease, but also responders and non-responders to medications, right? And I think, to me, that's exciting as we have more and more molecules that might be disease-specific. If we understand the genetics and the epigenetics, we'll start to be able to really customize our medications and our strategies.
Yeah, so great question, Nadir. So the question is again about how is the kidney on a chip, and maybe you want to extend and paraphrase organoid model systems, how are these more emerging technologies which are trying to recapture some of the organ's complexity in a more controlled fashion in vitro in the lab? How are they going to actually intersect perhaps with these genomic methodologies? How are they going to intersect, say, with genetic risk variants? Now we can all get genotyped or get 23andMe done. And then how is that actually going to intersect with how we think about disease susceptibility, adverse reactions, and potential response to treatment? And so I think really that's where this is slowly coalescing in nephrology. This is why it's an exciting time. And so I think the organoid systems, which I'm using loosely for both the actual organoids and the chip systems, are powerful, but we have to also understand that there are limitations. They're not the complete organ, and they sometimes don't reproduce all the features of it. But that said, especially when we think about, say, tubules on chips, it's an extremely powerful system to understand, say, adverse drug reactions. So we can actually genotype them, especially for a lot of the cytochromes that can be variant, and we can actually model adverse reaction as measured by tubular injury in vitro. Okay? And so then we can ask the one step before, in terms of thinking about epigenetics, how are these different cytochromes perhaps controlled in a differential fashion in tubules, right? Can we actually then use that information to predict whether a cell is going to respond poorly or better? So those are some of the questions we're interested in. In collaboration with an organized researcher, Bino Friedman at the University of Washington, he's very interested in polycystic kidney disease. And so they've actually taken stem cells derived from patients with polycystic kidney disease. They can make little tubular cysts in vitro. They'll actually puff up like a balloon, making cysts. And so he's actually done drug screens to identify which compounds may actually reduce that cystogenesis. We don't know exactly what's happening in terms of their enhancer landscape. So this is a project that we want to participate on together, right? To understand what does this mean in terms of reprogramming these cells. Why do some cells form cysts, but not others, right? So if we could separate the cyst-forming cells from the non-cyst-forming cells, which have the same genotype, can we understand what makes them different? Or why do some respond versus not? So clearly the whole book, the whole range of kidney pathology from the very beginning to the end is in play here, but we need very carefully designed experiments to try to execute them.
Hi, it's Sean at VGH.
Hi, Sean.
Thank you for a very interesting talk. My background is obviously in glomerular disease, so I tend to always think about things through that lens. I guess my question is there a possibility of superimposing a three-dimensional structural specific location of the epigenomic changes within a glomerulus as opposed to lumping the entire glomerulus together? And the reason I ask that is that I know that there's been some technology developments in transcriptomics to be able to look at specific areas of a glomerulus expressing different gene levels, and we have several diseases that presumably have very homogeneous inciting immune factors like ANCA vasculitis or IgA nephropathy, but seem to have very focal and segmental responses, you know, in the actual kidney tissue. And so is there something different about the epigenomic control in areas that seem to respond with injury versus other areas that seem to not respond with so much injury, despite a homogeneous immune event that started it all?
So that's a great question, Sean, about heterogeneity. And that, again, is reflective of how the field is going, trying to understand things at a single cell resolution. And I would say, actually, to your point, also at a spatial resolution as well, especially when we think about the glomerulus and the biomechanics of the structure is going to be very different one place to another. So that's clearly an area we want to go into. The technologies are eventually getting to the level of rigor that we can execute them in glomerular cells. We're not there yet, but that's definitely on the to-do list. Great question.
F1.
Yes.
Hi there, thanks. This is going to be a very non-elegant question compared to Sean's, but since you brought up 23andMe, so I had a patient yesterday who had just a slightly reduced GFR, that was it. So I gave him the whole spiel and watching, and then he sent me his 23andMe, and he had a factor that said, you are likely to have a reduced GFR based on this population study. It was RS1711437, that's what he had.
Okay. Well,
so I took a read,
yeah,
and you know, in a population, they did have reduced GFR. So I guess my question is, how do we educate ourselves as nephrologists about what the outputs, the renal-specific outputs of these home kits are, so that we can be a bit more elegant when we answer the questions from our patients?
Yeah, so that's a great question. I mean, so basically what that is showing is the result of kind of like a GWAS study, right? A genetic association study saying that that particular point in the genome is more likely seen in patients with elevated GFR versus normal GFR. And so the methods I was showing you today are at least the first steps to try to identify how, not the where, but how it might be actually occurring and which cell types might actually be contributing to that phenotype. And so I think one thing that we can talk to patients is that, you know, these alleles or these variants that are being annotated in 23andMe are really at a population level, right? And so we have to then understand what does that mean in the particular constellation of risk variants that that patient has, because that thing is not acting in isolation, right? What is happening in terms of, say, environmental risk factors that that patient may have on board? And then also, then how is that particular cell going to actually reflect that risk as well? So that's, again, a complex phenotype that we're trying to explain. But I think what you can tell them is that we're understanding a lot more about how these genetic risk pathways actually are intersecting with actual mechanisms. And I think as we are on that upslope and up-ramp of understanding mechanisms, we may have better opportunities to try to intervene, prevent, or at least slow down progression.
Is there a question in Victoria?
Hi, Ram. It's James Land from VGH.
Hi.
Hi. I really enjoy your talk, and I look forward to discussion with you and Paul when you come over to VGH in an hour or so. I'm coming from the literature of kidney transplantation, and as you know, there's a lot of cell-free DNA assays, different vendors.
Mm-hmm.
But I feel like your assay, your approach is much more elegant because, in a way, in a non-transplant setting, you have to define the signals of interest first, whereas in transplantation, you can just say this is donor-derived and then set a cutoff. What's been done recently, as you probably know, is they set a cutoff of 1% to define antibody-mediated rejection, which to me is very non-specific. Mm-hmm. And I would actually welcome your thoughts, and I think this could be a collaboration where we think about applying your approach to define specific signals that are for different types of rejection and different injured states in transplantation.
Thank you. Yeah, great question. And so this is also, I was chatting with the fellows at dinner yesterday about cell-free DNA as it's already made inroads into the kidney field, and as was mentioned, it's really made inroads into the transplantation field. Personally, I don't think it's that useful, but for many of the reasons that you have said so far is that it's not sensitive or specific enough. That particular methodology is leveraging the fact that there are probably SNP variants between the donor organ and the recipient that can be leveraged to identify where the DNA is coming from. Is it coming from the injured allograft, or is it coming from the host, right? But they're only using about 200 or 300 loci across the genome to base that inference. Versus the maps that we're showing here, we're potentially having thousands of these regions across the genome, and we're not relying on SNP variants from a donor to recipient. So really, we're taking the approach more of, can we identify which cell type is being injured, whether that's a tubule cell, whether that's an endothelial cell, to actually localize it to the type of injury. And we're hoping that it is a more sensitive and specific marker, but that still remains to be tested, road tested. That's, and certainly I think we would be very interested in collaboration on that front.
Well, thank you so much. I think this is a fantastically stimulating way to start January. I think that nephrology really is back because there's so much going on in this field, both at the molecular level as well as with new therapeutics. So it's really an exciting time. So thank you very much for coming. And I hear the Jeopardy went quite well yesterday, and the fellows did not embarrass themselves, and they knew enough pathology to be able to hold their own. So
always learn more.
That's right.
We're all learning.
Yeah, thanks again.
Thank you again, Andrea.